Good afternoon, viewers. Today in the lectures in EduSet network, we have in the lecture series in Immunology, we have a lecture on History of Immunology. A very interesting topic. I, Dr. Hardeep Kaur from Ramjas College, University of Delhi, will be delivering a talk on this topic. Now, as you know, History of Immunology is a very interesting one. Immune system is something which we are not aware of as such. It's only when something goes wrong that we feel that something is wrong with us, There are pathogens around us. Pathogens could be microbes, could be viruses, could be plant antigens, any kind of microbes, viruses, fungi, multicellular organisms, microorganisms, anything could be parasites. They all are pathogens which are around us. They are inflicting our body at all regular intervals, but it's our immune system who protects us. That is what is important. But it's only when something goes wrong that we come to know that something is wrong with the body. Till that time, we are not aware that there is such a good function, such a good system which is acting in our body. So today we are dealing with history of immunology, how immunology came into being. What were the first experiments which were done by scientists probably in 2000 BC or before that. So, coming to immunology, immunology stems from a Latin word immunis, which means exempt. In English, it means protection from diseases. It was seen in historical times that people who got inflicted with certain diseases became immune to those diseases. Whenever they came in contact with those same pathogens again, they were found to be exempted from getting a disease again. They were willing to take care of the people who were inflicted with the same disease, and they never got an infection again. Hence the term immunis came, which in Latin means exempt from that disease. The term immunity was first used in 1775 by Van Swieten, a Dutch physician, as immunitas to describe the effects induced by an early attempt at virilization. Virilization is something similar to variolation, which means inoculation against smallpox using material from a smallpox lesion. Now, going back into history, as early as 2000 BC, diseases and epidemics were recorded in Egypt. Initially, it was considered that a disease is a punishment by God. A person who has got an evil in his heart would be inflicted by a disease, but the person who hasn't got an evil heart would not contract that disease. And a person who has made good with God, who has come back to his senses, would be cured of the disease, and he would be able to live his life. Otherwise, he might die of the disease. So it was considered a kind of punishment by God. Under that scenario, the science of immunology did not flourish too much with that thought in mind. According to Thucydides is a great historian of Peloponnesian War. In 430 BC, a plague inflicted Athens, and it swept throughout Athens, and those individuals who survived the attack were able to take care of the sick. They did not experience plague again. This was the first documented evidence of immunology, of something. See, immunology has been there for ages immemorial, but nothing was documented. This is the first documented evidence which shows that people were thinking about the immune system. Now, coming again to historical perspective, variolation or virilization, which was initially said, it's an obsolete process of inoculating a susceptible person with material taken from a vesicle of a person who has smallpox. The origins of inoculation against smallpox in China is known. Initially, it's been known that there was a province, a southern province in China which was called Sichuan, and references has been made to a temple called O Mei Shen, where there used to be some Buddhist and Taoist alchemists. Taoism was the religion which was followed in China in probably in 10th century. And near this mountain, this is a mountain temple, Omin-shen, and under the caves of Omin-shen, Buddhist and Taoist alchemists used to live there, and they were considered to be following immunization or variolation, the process of variolation. But this was not known that they had been following it. This technique actually came into being first time into public attention when the eldest son of Prime Minister Wang Tan died of smallpox. Wang Tan was very unhappy, very sad, so he didn't want his other members of his community, of his family to die of smallpox. So he asked for all the magicians, for Buddhists, for Taoist alchemists to come and give a cure to other family members so that they don't get inflicted with smallpox. So one of the monk, who's been called a holy monk, whether it was a lady, nun in that case, came and she actually followed this technique of variolation, where the material was taken from a vesicle of person who was inflicted with smallpox and put onto a healthy individuals. So this was the first case of variolation. In his book Daozen Zinfa, published in 1549, Chinese author Wen Quan made clear reference to the process. So it's quite clear that probably in the 14th century or 15th century or 16th century, the process of variolation was taking place. Inoculation from smallpox does not appear to have been widespread in China until the reign era of Longqing Emperor during the Ming Dynasty. Description, as you can see in the picture, of this practice are recorded by Yu Chang in his book Miscellaneous Ideas in Medicine of 1643, where you can see a needle has been used to take out the material from the pustules of smallpox. A person suffering from smallpox pustules. His pustules, the material is taken and is used to inoculate another person, a technique which now is called variolation. During the 17th century, the practice from China was slowly introduced into neighboring countries along with the Silk Road, and it was in Turkey that it came to the attention of Europeans. English merchant John Lister reported the Chinese method to the Royal Society in 1700. In pre-colonial India, tikka or dot would be made on the sole of the foot by tikkadars. It was nothing but another process of variolation where a material of smallpox pustule is put into a healthy individual. As you can see in this slide, it's an Indian variolation vial containing live smallpox virus. Now, as I was just telling you, the process of variolation or the process of early giving immunity through variolation actually started in China, and later on through the Silk Route went to Turkey. Now there was this Lady Mary Wortley Montague, who first discovered inoculation for smallpox while she was in Constantinople. This lady was a beautiful poetess, and her husband actually was a British ambassador to Constantinople, which is now Turkey. And for a 2-year tenure from 1716 to 1718, she stayed in Constantinople. While being in Turkey, she came across people following this variolation very actively. She had a daughter and a son. She inoculated her daughter with the technique of variolation through smallpox material, and later when she returned to Europe, she inoculated her son also with the same in 1721. She found out initially there weren't many takers, people ridiculed her, there weren't many takers, and she was made to, people did not follow her. But later, when this was taken up by the royal people, the British, the Queen and the King, they took this variolation process, then the mass people started following this process. 6 prisoners sentenced to execution and 6 orphan children were variolated to test the theory. So it almost took probably in 1780s that the first mass variolation took place in Europe. When exposed to smallpox, all these people, whether the 6 prisoners or the 6 orphan children, they all survived. The royal families after that, as I just said, began doing the process of variolation, and the practice effectively spread to Europe. As Lady Mary Wortley Montague says, the smallpox so fatal and so general amongst us is here entirely harmless by the invention of ingrafting. That was the term it was used that time. It was not called variolation, it was called ingrafting. There is a set of old women who make it their business to perform the operation every autumn in the month of September, when the great heat is abated. You must understand, during this month it is generally an outbreak of smallpox in Europe, because it's a heat and it's a change of season. People generally get afflicted with smallpox. So during that time, these ladies will come up and they will do this operation of variolation. People send to one another to know if any of their family has a mind to have the smallpox. They make parties for the purpose. So technically this is in her own words, where Lady Montague is actually describing what practice was followed in those days. By the second half of 18th century, Europe was being ravaged by smallpox epidemics. By this time, in rural England, it was noticed that women who milked cows were frequently spared clinical smallpox disease. Now, it was seen, it was actually, if you consider this, this knowledge was already widespread, that people or the maids, the cow maids who tend to cows and who happen to have cowpox virus, they are generally protected from smallpox disease. But this, and people had been following it, as I will just tell you after this Edward Jenner slide, that there were people who were already following this, but there has not been documented widely. In fact, Edward Jenner, who was born on May 17, 1749 in Berkeley, Gloucester, England, he as a teenager, while learning to be a physician, he heard a young farm girl telling doctor that she could not contract smallpox because she had once had a cowpox. Now, today we know that why this happens. So, because cowpox and smallpox are both caused by vaccinia, one by vaccinia minor, the other by vaccinia major respectively. It is because of the similarity in the pathogen that people who get afflicted with the cowpox do not get affected by the smallpox virus. This started him to think about making a vaccine. In 1796, Sarah Nelms, a local milkmaid, contracted cowpox and she went to Jenner for treatment. Jenner found it as an opportunity to test his theory. He inoculated James Phipps. It's a very famous experiment in the history. He inoculated James Phipps. You can see the hand of Sarah Nelms in the picture. This is the hand of the cowpox pustule which she had developed, and through this he took a material and inoculated James Phipps, the 8-year-old son of his gardener, with material taken from cowpox lesion on Sarah's hand. You must understand, this is a very normal technique nowadays. What he is doing is, he is taking a very weakened strain, or rather a weak strain of vaccinia, putting it into James Phipps, and after some time he developed a mild fever. After some time he recovered from the fever. A month later or few days later, he again inoculated James with this time with smallpox material. Ordinarily, inflicting somebody with the smallpox who has not been vaccinated or variolated before would have led to a full-blown smallpox infection. But here in case what happens, James Phipps was able to recover. In fact, he did not develop any symptoms. So, he was totally protected from smallpox disease. In fact, as out of gratitude, Edward Jenner gave a house to James Phipps. This house was just next to the house of Edward Jenner in the same lane where his house is in Gloucester, England. So, this was out of gratitude that he gave him a house also. Jenner wrote a paper in 1798 explaining his experiments and wanted to report his first case study in the Transactions of the Royal Society of London. However, his study was rejected. He then went to London to demonstrate his theory. No one submitted to his vaccination theory. Discouraged, he came back to Berkeley. In 1801, Jenner published the first paper, The Origin of Vaccine Inoculation, describing how cowpox virus was prepared and used to protect healthy persons against smallpox. Now, whether Jenner was the first one to follow this technique of variolation, as I just told you, the variolation technique had been there since ages immemorial. People had been doing it, but there is no documented evidence for it. However, it's been, although Edward Jenner is known as father of smallpox vaccination, he dedicated his entire life, money, reputation for preparing a vaccine, but in 1774, that is 2 decades before Jenner's experiment on James Webb, a farmer called Benjamin Jesty vaccinated his wife and children at Minster in Dorset. Now, Jesty, the Benjamin Jesty, who was a farmer, he had actually heard a lot of variolation. So, what he did, he just, when he realized, he had 2 mates who had contracted cowpox, and those mates told Jesty that because they had cowpox, they would not have smallpox, and that, as I was telling you, that it was quite followed in those days. Now, Jesty, what he did, he realized when his cow became afflicted with the cowpox, he took his family to the farm and with the needle took the material from a cowpox pustule and inflicted it onto his wife and to his children, and he kept mum about it. He did not want to spread it, but unfortunately for him, his wife developed fever. So, he had to call a physician. He did not want the neighbors to, he never wanted the neighbors to know about it, but unfortunately for him, his wife developed fever, the physician was to be called, doctor was to be called, and it became a common knowledge what he did. And after that, he was highly ridiculed, he was hooted wherever he used to go, he was pelted with stones whenever he used to go to the market. So, he had to actually change his living place, and it's been written, the new place where he went to live, it's been written on the gravestone. The entire story about his life is written on the gravestone of Benjamin Jesty. So, definitely Jenner was never the first one who developed, who utilized variolation, but yes, Jenner was the one who actually popularized variolation. Now, from now came the word vaccination. It's a Latin word from vexa, means cow. In 19th century, the French scientist Louis Pasteur experimented with antigens to combat anthrax and rabies and called these antigens vaccines in Jenner's honor. Now, there is a story about Louis Pasteur. Louis Pasteur had cultured the anthrax bacillus in his lab. So, on one of his vacations, the vacations became long enough. When he came back, the anthrax which he was having, that became very weakened. So, he just tried that anthrax bacillus on the fowls which he was having, the new fowls which he got procured, and those fowls developed a very mild disease. So, he was quite surprised, because till now, whenever he used to be inoculating the fowl with the anthrax bacillus, they always used to die. This was the first time. So, what he did, he again subcultured the anthrax bacillus, and this with virulent and good anthrax bacillus, he now again wanted to inoculate a fresh batch of fowl, but for his disadvantage at that time, he could not get a fresh batch of fowl. So, he had to do with the same fowl who had recovered from the disease. So, again to his surprise, this batch of fowl did not develop the disease at all. So, this was the first proof which he thought that probably a weakened or an attenuated pathogen can be used as a vaccine. So, this is how he coined the term vaccination in honor of Jenner, where vexa means cow. Later on, in his very famous experiments in Polyleaf Fort, he actually again inoculated a batch of sheep with anthrax bacilli. This anthrax bacilli was very virulent, and he found that all those sheep who had previously vaccinated with anthrax did not develop this disease, while while the batch of sheep who did not had a vaccination with the anthrax, they developed the disease and they died. So, this were all experiments. First, the experiments of the chicken or the fowl for their cholera, and then with the sheep for anthrax bacillus. Later, he used the same technique to treat a boy against the rabies pathogen. Nowadays, we use the word vaccination to describe a procedure in which a patient is inoculated with an antigen to produce immunity against a disease. So, coming back to Louis Pasteur and Koch and the germ theory. In 1875, Robert Koch, a country physician with no formal scientific training, inoculated the ear of a rabbit with the blood of an animal that had died of anthrax. The rabbit died the next day. He isolated infected lymph nodes from the rabbit and was able to show that the bacteria contained within them could transfer disease to other animals. He discovered the tubercle bacillus and tuberculin. Detailed tuberculin skin test, that is DTH, fetched him a Nobel Prize in 1905. Louis Pasteur, as I was telling you, he began his studies using chicken cholera bacillus. Pasteur later worked on anthrax and rabies and developed the first viable vaccine for anthrax and rabies. He also developed pasteurization, a process by which harmful microbes in perishable food products are destroyed by heat without destroying the food. On July 4, 1886, 9-year-old Joseph Mister was bitten repeatedly by a rabid dog. Pasteur treated him with his attenuated rabies vaccine 2 days later. Mister survived. Joseph Mister later became a gatekeeper for the Pasteur Institute. In 1940, when he was ordered by the German occupiers to open Pasteur's crypt, Joseph Mister refused and committed suicide. So, he actually became a gatekeeper to the laboratory of Pasteur and protected with all his might till he died. Later in 19th century, Emil Adolf von Behring, a student of Koch with Kitasato and Wernicke, discovered antitoxin for diphtheria and tetanus and applied it as a therapy. They were awarded the first Nobel Prize in Physiology in 1901. Before going on further to this, I would like one more thing to tell about Louis Pasteur and Koch. Although Louis Pasteur and Koch have been credited with finding the germ theory, but they had a bitter rivalry between Koch and Pasteur. In fact, in one of the conferences where Koch and Pasteur both were present, Pasteur gave a lecture. So, once Pasteur finished his lecture, Koch was to deliver his lecture. So, he started off by saying that Monsieur Pasteur was invited, so that was the reason that I accepted the invitation. But after hearing the lecture, I found that Monsieur Pasteur did not give anything new. So, I am highly disappointed. This was the kind of rivalry. They used to actually pull each other's legs in each and every conferences. So, but nevertheless, their contribution in the field of immunology is immense. It is because of them that the current vaccination and all those important immune system things we have come across. We would never have been here had it not been because of Koch and Pasteur. Later, Paul Ulrich in 1850, who was born in 1854, he developed a series of tissue staining dyes, including that for tubercle bacillus. He worked with Koch and developed antitoxin to diphtheria. He gave a side chain theory of antibody formation. He says that antibodies are kind of receptors and they are bound in a lock-and key manner with the antigen. That means antigen binds to the antibodies that are present as a receptor on lymphocytes in a lock-and key manner. That means just like for every lock there is a specific key, for every receptor there is a specific antigen, or vice versa, for every antigen there is a specific receptor and antibody. In 1908, Paul Ulrich shared the Nobel Prize with Metchnikoff. Now, talking of Elie Metchnikoff, he was an embryologist who studied starfish development. In fact, the cellular immunity or the cellular immunology, he was the person who actually founded the cellular immunology. He was working on starfish larvae and he found out, he was looking at the digestive system of the starfish larvae and he found out that whenever he was putting some dye inside the starfish larvae, there used to be cells who would come and pick up all that dye and would remove the dye from there. So, those cells were phagocytes, which he called them as phagocytosis, where phagos means to kill or to devour, right. So, he shared the Nobel Prize with Ulrich in 1908 and he actually gave the cellular concept of cellular immunology, saying that probably these cells, phagocytes and all, are the first line of defense, which is actually true. Phagocytes are actually the first line of defense which take care of any pathogen which enter our body. Later on, Sir Frank McFarlane Burnett, who was trained as MD, he had an important work on influenza. The discovery of an influenza viral enzyme with the specificity for particular forms of uremic acid, which is now used for detection. He also gave the clonal selection theory to explain tolerance. Initially, two theories were formulated. One was the instructional theory, the other was a selection theory. The selection theory said that it is the antigen which comes and selects its specific receptors and then bind it in a lock and key manner. The instructional theory says no, just like protein has a kind of lot of variability or a very malleable or a very that kind of a structure which can change. So, similarly, the antigen can come inside, the receptor which is a protein molecule can change accordingly and make a good fit with the antigen. So, the instructional theory actually said it is the antibody or probably the receptor in those times, receptor which makes itself adaptable to the antigen. All these theories were later removed and a new theory thanks to Burnett came, which was called the clonal selection theory. According to the clonal selection theory, the antigen comes inside, finds its appropriate receptor, which is an antibody in this case, let us say, it then binds to it and then it gives an instruction to the lymphocytes to make clones of the same. So, that a particular lymphocyte which is producing a particular receptor or an antibody will be having a clonal expansion, many of its clone will be produced. So, numerous number of receptors would be given off and these receptors will bind to the antigen and hence the antigen will be removed. This was the theory given by Burnett called clonal selection theory. This fetched him a Nobel Prize in 1960 for the discovery of acquired immunological tolerance. This was followed by Rodney R. Potter and Gerald Edelman. In 1972, they got Nobel Prize for the discoveries concerning the structure of antibodies. We all know today the structure of antibodies, the Y-shaped structure with the variable and the constant chain and the light chain and the heavy chain. That structure was for the first time given by Rodney Potter. Baruch Benacerov, George T. Snell, Jean Dosset, they discovered genes that regulate immune responses. Nowadays, we call them as histocompatibility antigens. They got Nobel Prize in 1980 for their discovery. These histocompatible antigens have a major role to play, specifically whenever the grafting has to take place. We now know what tissue typing is. The tissue typing is only possible because of the knowledge of the histocompatibility antigen and thanks to Benacerov, Snell and Dosset for the discovery. Neil K. Journey and Mistel and Kohler, they got Nobel Prize in 1984 for theories concerning the specificity in development and control of the immune system. They talk about the regulation and the discovery of the principle for production of monoclonal antibodies. They were the ones who gave the hybridoma technology. They received Nobel Prize for monoclonal antibody production in 1984. Now, these are a list of Nobel laureates in immunology and closely related fields. As you see, it started off in 1901 with Emil von Behring getting a diphtheria antitoxin, their work for diphtheria antitoxin, the first Nobel Prize, followed by Paul Ulrich, Elie Metchnikoff, Baudet, Burnett, Middawer. The list is endless. These are some of the Nobel Prizes given in immunology to the related fields. For example, Karl Landsteiner was the first one who gave the theory of blood group typing. This was followed by many of them, then Rosalind Yellow who gave radioimmunoassay in 1977, Donald Thomas 1990 for bone marrow transplantation. All these prizes This is all the compilation which has been taken from a paper called Glittering Prices by Peter C. Doherty in Nature Immunology in 2010. Now, as I would just like to end my discussion by saying, scientist is not a person who gives the right answers. He is one who asks the right questions. Let's use our stories to encourage listening to one another and to hear not just the good news but also the pain that lies at the back of a lot of people's stories and histories. They must have endured a lot of pain. Many of them must have been ridiculed. Many of them been told that no, please, this doesn't, it doesn't happen like this. It's all because of, as I told you, it all started with people saying that disease is because if you have angered God, you know. With that mentality in mind, coming to the point where we now know why diseases take place. We now know the vaccination. We now know how to cure the diseases. So, all these things are very important and we must understand there is a long stories behind each discoveries. To go into and unravel those historical movements, the historical perspective is very important for us scientists to understand the pains that has been endured by all the scientists. I would like to finish my lecture by saying that we all must hear, must read, must go through the history of these scientists, how these discoveries have came into being, how they happened, and then we will be able to appreciate how much work has gone into these discoveries. Thank you very much.